For the introduction part, this research paper from NCPI presents a comprehensive bioinformatics analysis of the HIV-1 envelope gene with a focus on its structure, functional domains, and implications for viral entry and pathogenesis using data from full-length, unligated GP120 and GP160. We explore how the ENV sequence interacts with cellular receptors and identify key conserved motifs that influence infection dynamics. The HIV-1 ENV gene spans 2,568 base pairs, encoding the GP160 precursor, which is proteolytically cleaved into the surface protein GP120 and the transmembrane protein GP41. Sequence data for this study are available under accession number GI9629363, which corresponds to the GP160 precursor. The closest known relative of HIV-1 is the simian immunodeficiency virus from chimpanzees.
Now, let's take a look at the bioinformatics tool used in this study. Since the method applied is the position-specific scoring matrix or PSSM, the tool used is WebPSSM, an online platform that predicts HIV-1 co-receptor usage based on NVV3 sequence data. WebPSSM is a web server type with its function is to predict HIV-1 co-receptor usage, known as tropism, for NVV3 sequence data using the PSSM method. For the key application, it is used for co-receptor usage analysis, distinguishing between CXCR4 and CCR5 tropism, and also support treatment decision involving CCR5 antagonists. WebPSSM is freely accessible online through the WebPSSM server hosted by the Fred Hutchinson Cancer Center. Researchers and clinicians can simply upload HIV-1 NVV3 sequence data to obtain co-receptor usage predictions. Now, let's look at the advantage and disadvantage of using WebPSSM. For the advantage, first, WebPSSM offers high specificity, providing reliable prediction with a strong ability to exclude false positive in co-receptor usage analysis. Second, it provides a balanced sensitivity and specificity, making it useful for both clinical and research application. Third, it shows competitive performance by performing as well as or better than other tools in predicting co-receptor tropism. Lastly, WebPSSM is user-friendly. Being a web-based tool, it requires no complex installation and offers easy input and fast predictions. However, there are some limitations to this tool. Firstly, it is limited to the V3 loop sequence, meaning that it focuses only on this region and may miss other factors influencing co-receptor usage. Secondly, it has reduced performance with incomplete sequences, so shorter or gap V3 loop sequences can lead to inaccuracies. Third, generating PSSM profile can be time-consuming for large dataset, as it can be slow and resource-intensive. And lastly, WebPSSM is less effective for detecting minor variants. It may miss low-frequency non-R5 variants in mixed viral population, which are better identified by phenotypic assays.
First, for analytical method, which is method explanation, PSSM, or position-specific scoring matrix, basically represents a mathematical representation or method. It is a metric that represents evolutionary diversity and conservation by quantifying the probability or score of each nucleotide or amino acid occurring at each place in a sequence alignment. It is often used to classify sequence according to patterns, anticipate functional locations, and evaluate sequence motif. Then, for integrative analysis of the HIV-1 ENV V3 loop, which is structure, tropism, and evolution. First, sample collection and sequence extraction. Viral RNA or proviral DNA is extracted from patient samples, for example, sera or peripheral blood mononuclear cells. The ENV gene region encoding the V3 loop is amplified using polymerase chain reaction, which is PCR, and sequenced directly. So, for explanation, we collect patient blood samples. From this, we extract the virus genetic material. Using PCR, we copy a specific region called the V3 loop and then sequence it to read the genetic code. Second, sequence alignment and quality control. The V3 loop sequences, typically 35 to 37 amino acids, are aligned to referent HIV-1 sequences to ensure correct reading frame and to identify conserved and variable residue. Homology between genomic RNA and proviral DNA sequences is confirmed to validate sequence accuracy. And for explanation, we align the sequence with referent HIV-1 sequences to check for accuracy. These steps helps us to find any errors and make sure we are comparing the right parts of the virus. Third, sequence feature analysis. Critical amino acid, residue, and motif in the V3 loop are identified that correlate with viral tropism. For example, the presence of positively charged residue or specific motif such as QR, GPGR are linked to lymphocytotropism or macrophage tropism 14. And for this step, we look closely at the V3 loop to find special patterns of features. Certain amino acid or charge can tell us how the virus enter human cells. Fourth, prediction of co-receptor usage. Using bioinformatics tools, for example PSSM or Genome, the V3 loop sequence are scored based on non-sequence pattern associated with CCR5 or CXCR4 usage. This algorithm analyzes sequence variability, charge distribution, and motif presence to predict viral phenotype, which is R5 trophic, X4 trophic, or dual trophic. And for this explanation, using online tools like Genome or PSSM, we predict whether the virus use the CCR5 or CXCR4 receptor. This is important because it affects how the virus spreads and which drug might work best.
Next, HIV-1 co-receptor prediction flow diagram using PSSM method. First, we need to get a patient sample collection. Next, we need to do HIV-1 V3 loop sequence data. Next, RNA or DNA extraction. Next, PCR amplification of NV V3 loop. Next, sequence quality control and alignment. Next, we need to do PSSM scoring using web PSSM using alternative SVM or RF. Next, bioinformatics prediction of co-receptor usage. Next, phylogenetic and clustering analysis. Next is structural modeling of V3 loop. And the last one is clinical correlation and therapeutic guidance. Next, method application. First is research on viral pathogenesis, identifying amino acid connection and sequence patterns associated with co-receptor preference reveals molecular processes of viral interest and tropism alterations leading to disease progression and immune evasion. Second is clinical decision support. The method predicts co-receptor tropism using patient-derived virus genome, allowing for more tailored antiretroviral medication and improved treatment success. The last one is epidemiological surveillance, large-scale study of HIV-1 sequence from various population and subtypes aid in tracking the evolution of co-receptor use patterns, which inform public health initiatives and help to understand viral adaptability.
For conclusion, bioinformatics tools and methods such as PSSM based analysis play an important role in understanding HIV-1 co-receptor usage and viral evolution. They provide valuable insights that support good clinical decision making and research, while also highlighting the need to consider tool limitation and sequence quality for accurate results.